one of the most important things that needs to occur during cell division is to copy all of your DNA inside and distribute the copies of DNA to the daughter cells that will form from that. So the word that we use is genome. Genome is the term that encompasses all of the genetic material in that organism. That is that organism's genome. Now, if you're a prokaryote, you have one chromosome most of the time, and that chromosome is circular. Now, eukaryotes have multiple linear chromosomes instead of circular, so eukaryotes do have vastly larger quantities of DNA they need to copy. Now, we can find DNA in places other than the chromosomes in prokaryotes and eukaryotes. For example, we have plasmids. We discussed plasmids before. They are smaller circular pieces of DNA common to prokaryotes. Mitochondria and chloroplasts also have their own DNA that needs to be copied. But DNA in plasmids, the mitochondria and chloroplasts, they sometimes they get copied when the DNA, the rest of the chromosomes are being copied during cell division. Other times, if we need to copy plasmids, mitochondria, or chloroplasts, then they can also be copied outside of cell division just as needed when copies need to be made. For eukaryotes, our DNA is incredibly complex. I mean, it's complex in prokaryotes too. It's just more complex in eukaryotes. So first of all, we've learned the idea of chromatin. Remember from previous chapter that chromatin is the state of DNA in a non-dividing cell. Chromatin is not just DNA all by itself. Chromatin is a mix of DNA wrapped around various types of proteins. So looking at this picture here, we see the standard double helical DNA right here. But then what happens in yellow are some of the proteins that DNA is wrapped around to form chromatin. A really common type of protein that DNA is wound around to form chromatin are proteins called histones. The way this works is you'll get several histone proteins that clump together to form a core. And then once we form that, the DNA is then wound around that histone core a couple of times. So in some places, our chromatin looks like this little beads on a string type of necklace. Every little bead, which is made up of the histones plus the DNA, that is called a nucleosome. Nucleosome includes both the DNA and the protein, the histone proteins that it's wrapped around. So we have these nucleosomes, one after the other. What can happen in other parts of chromatin is the nucleosomes get wound up into a coil that looks something like this. This is referred to as a chromatin fiber or a solenoid. And chromatin can look like either one of those, individual nucleosomes one after the other, or the solenoid form. And even in one piece of DNA, one molecule of DNA, parts of it can look a little more relaxed, like just the nucleosomes. Other parts are wound a little more tightly, as in the solenoid shape. Okay, so that is the idea of chromatin. Now, what will happen during cell division is the chromatin is still too long and stringy to be able to kind of move around the cell as the cell needs to divide. So what happens is the cell will take the chromatin and condense it to a structure that is much more compact. It's very short but very thick, and it's, as you kind of wind the chromatin up into this chromosome, highly condensed form of DNA, it is much easier to move the DNA around during cell division in the chromosome form. So going back to this picture here, the chromatin gets looped around, and then the loops get kind of smashed together or condensed, and then that gets condensed even further to form the fully formed chromosome. So technically, our DNA isn't in the chromosome state unless the cell is dividing. All the other time, it exists as the chromatin that must be condensed into a chromosome, again, only during cell division. And as soon as the cell is done dividing, it will decondense the chromosome back into chromatin so it can start using the DNA again. Because as the chromosome state, the DNA is way too compacted. The genes are inaccessible. We can't really use them to make what they code for. It's only when it's in the chromatin state like that. So looking at this picture of a chromosome here, chromosomes are fairly complicated. This is what the chromosome looks like after we have made a copy of every piece of chromatin or every piece of DNA in the cell. Okay, so looking at this, this piece of DNA and this piece of DNA, these are identical copies. We used to just have this piece of DNA here, but then when the cell started the process of cell division, it made an identical copy of this piece of DNA. And here's a copy of it. And then, of course, it was chromatin, but each identical piece of chromatin was condensed into something much shorter and much thicker to form the structure here. So even though this structure here is made of two identical separate pieces of DNA, those pieces of DNA are held together. And because they are physically connected, this is considered one chromosome. So what you see here, this is one chromosome. Each condensed piece of DNA on the right and on the left here is a chromatid. And since these two chromatids are identical to each other, they are what are known as sister chromatids. Okay. Now, the glue that holds the two sister chromatids together in this one chromosome are made of proteins called cohesins. Cohesin proteins are cohesive. They hold things together. Where the two sister chromatids are held together very tightly, the chromatids get a little thinner, sort of like a waist, and each waist of the chromatid is known as a centromere. Central means central, mere means part. So the center part, the thin waist part here, that is the centromere of this chromosome and the chromatids. Okay? And all the centromere is just the area, the region where the two chromatids are held together very tightly, and they're a little thinner there. The centromere doesn't have to be right in the middle. It can be shifted off to one side or the other, or really close to one edge or the other. But it's the centromere. Okay, other parts. We have proteins on the outside of each centromere here and here. These proteins are referred to as kinetochores. The function of a kinetochore is to be an attachment point for these microtubules here to connect to the chromosomes. So if we want to move chromosomes around the cell during cell division, this is how we have to do it. We take these microtubules, this type of cytoskeleton, these microtubules, and we hook them to the kinetochores on each side. And those microtubules can push and pull and drag the chromosome around the cell during cell division. We need kinetochores to attach the chromosome, or to attach the microtubules to, because we don't want to risk damaging the DNA by connecting the microtubules directly to the DNA here. We could damage it, uncoil it, or simply tear the DNA. So kinetochores are much tougher, stronger kind of a protein. They're used to drag the chromosome around, sort of like a handle. Okay? So this is one chromosome, and at this point, this one chromosome is made of two chromatids. Here's an electron micrograph. This means a picture taken with an electron microscope, a very powerful kind of a microscope, and we can see this chromosome is really just condensed chromatin.